Welcome to the third week of class. In this lecture, we will learn more about the influence of physiology, the other field of study that contributed to the birth of psychology. There are three categories of studies we will cover in this lecture: decapitation studies in the 1700s, animal brain studies in the 1800s, and nerve cell studies in the late 1800s, early 1900s. Let's review the difference between philosophy and physiology. Remember that philosophy is the study of knowledge, and physiology is the study of the body, its parts, and how the body and its parts function. In the 18th and 19th centuries, decapitation studies contributed to the early days of psychology. Robert White was a Scottish physician, and he was one of the first individuals to study decapitated heads and share those results with the scientific community. He used Luigi Galvini's method, which is depicted here in this image. White inserted hot wires through the spines of decapitated frogs. When the frog's spine had been destroyed, there was no response to the hot wire. When the spine was still intact, the legs of the decapitated frogs actually moved. They retracted when White sent electricity through the wire. He concluded that the spinal cord is all that is needed for reflexes to occur, no brain necessary. Then Theodor Bischoff, a German physician and biologist, began to study human heads. Now don't worry, he didn't do the decapitating. He collected the heads after execution by guillotine. A guillotine is shown here in the example. He also collected the heads and embryos of a variety of animal species, including dogs, deer, and pigs. Those animals were relatively easy to get his hands on. He concluded consciousness ends at death. How did phrenology, a pseudoscience, contribute to the development of psychology? Franz Josef Gall is the father of phrenology. In 1819, he wrote that personality can be inferred from the bumps on your head. His ideas would later become known as phrenology. He felt the skulls of more than 100 family, friends, and colleagues. He concluded after his analysis that there are 27 faculties of the mind. Each one of these faculties is a function of the mind. The image on the screen depicts one of the earliest illustrations of the functions proposed by phrenologists. If you look right behind the ear, phrenologists said this area of the brain was in control of destruction. If you had unusual bumps behind your ear, Gall inferred that you were more of a destructive person than someone who doesn't have bumps behind the ear. Phrenologists believed the brain is very localized, that specific areas of the brain were in control of very, very, very specific behaviors. They also believed that everyone has the same set of 27 functions, but there are individual differences in the strength of each one. These faculties are inborn, they are innate, we are all born with them. The doctrine of the skull suggests that the strength of each of the 27 faculties depends on the bumps on the head. In addition to promoting their ideas about skull shape, phrenologists also invented tools to use in measuring skull shape, bumps, imperfections. This is an example here on the screen of a craniometer. It was designed to measure the distance between different parts of the brain and help determine the depth of indentations. What did they get right? Gall was correct that different parts of the brain are responsible for different functions of the mind. Different parts of the brain are responsible, at least partly responsible, for different basic human functions. But the map of 27 functions, the idea that the strength of these 27 functions depends on the shape of the skull, has not been supported by any research. Their reliance on anecdotal evidence, the notion that Gall felt the skulls of his family, friends, and colleagues, overlooked non-supporting cases. We have to look at those cases and try to make sure our theory incorporates those cases. Phrenology remained popular in the United States and in Europe until the mid-1800s. In the early 1800s, there were two individuals who were mainly responsible for spreading the word about this particular set of ideas. Johann Spurzheim, a German physician, and George Combe, a Scottish lawyer. Both of these individuals traveled around Europe and the United States, going to different community events, setting up a tent, and bringing people into the tent, feeling their heads, and then giving them a personality profile. Unlike Gall, Spurzheim said that faculties could be shaped by early experiences. He believed that genes were not the only determinant in personality, behaviors, thoughts. This idea that we have free will and we can make decisions about the people that we become, that was very much in line with the American values and ideals at the time. The United States was growing rapidly. More and more individuals from all different parts of the world were coming to the United States. My own relatives, the Quartons, came to Illinois in 1836. My great-great-great-great-grandfather wrote a letter back home to England that year describing the farm that he had purchased in Jacksonville, Illinois. Because of this alignment, Spurzheim's version of phrenology really caught on in the United States, and it was 1860s before the scientific community was able to convince the general population that there were better theories, better alternatives to the study of the mind. In the 1800s, physiologists conducted a variety of animal studies. Let's check some of those out now. Jean-Pierre Flourens developed a technique in the early 1800s that would prove very useful to the study of the mind. He was a French physiologist, and his work was the enemy of phrenology. The results of his studies provided evidence that disproved or poked holes in the ideas, the basic premise of phrenology. Flourens agreed that different parts of the brain have different functions, but Flourens, using his brand new state-of-the-art technique, was able to show that the functions proposed by phrenology were not quite right. He used the method of ablation. He surgically damaged brain tissue in rabbits and pigeons and observed the effects. When the cerebrum was damaged, the animal struggled with perception. It was difficult for the animal to see. It would run into things. It was difficult for the animal to hear. He would blast different sounds. The animals with cerebrum damage had difficulty hearing those sounds. When the cerebellum was damaged, movement was a problem. Not only was it difficult to walk, but simply standing still was a challenge for the animals. When the brain stem was damaged, as you can imagine, the animal died. Decades later, Pierre Paul Broca studied the brain of one of his patients who had motor aphasia. This patient was unable to speak. The only word this patient could say was tan, which is why the patient was named Tan. It wasn't his real name. The interesting thing is Tan could comprehend speech. You could speak to Tan and he would understand what you were saying, but he did not have the ability to form words and push those words out of his mouth so that you can hear them. After a natural death, Broca did an autopsy on Tan's brain and found significant brain damage. The brain you see here on the screen is Tan's brain. It is an image of Tan's brain. Broca concluded that the ability to produce speech is located in the area of the brain that experienced the most amount of damage. This area is now known as Broca's area. Francois Magendé was a French physiologist who worked primarily with puppies. What did he do to these puppies? He cut different parts of their spinal cords and watched what would happen. True story. When he cut the dorsal posterior root, it's a specific part of the spinal cord. When he cut the dorsal root, the puppies could move their legs, but when you pinched the puppy didn't respond. The puppy couldn't feel a pinch. When Magendé cut the other root, the ventral, the anterior root, the puppies couldn't move their legs anymore, but they squealed in pain when he pinched them. This observation led him to conclude that these two different roots were responsible for sending two different types of information to and from the brain. The dorsal root is the sender of sensory information, the ability to feel. So when he cut the dorsal root, the puppies couldn't feel the pinch. The ventral, the anterior root, is primarily responsible for movement, because when he cut that area of the spinal cord, the puppies could no longer move their legs. Today we call this principle in physiology the Bell-Magendé law. One of the most famous physiologists of all time, especially at this particular time in history, is Hermann von Helmholtz. He was a German physician and physicist. Many of his ideas, you can see, have a very strong foundation in the natural sciences. He researched sensation and perception, seeing, hearing, feeling. He also taught Wundt, the father of experimental psychology. It is from Helmholtz that Wundt learned many of the most basic techniques of the lab. One of the things Helmholtz studied was nerve impulse speed. He wanted to know how long does it take an impulse to go from your brain to, say, your toes. So he conducted one of the first reaction time studies in psychology to try to determine nerve impulse speed in both frogs and in humans. He tested frog muscles and gathered rates of about 300 meters per second, 90 feet per second. That's fast. When he tested humans, he found that the rates were twice that, 180 feet per second. That's equally impressive. He also studied visual perception and invented the ophthalmoscope. Along with James Maxwell, he found that they could create any color by mixing red, green, and blue, the three primary colors. They concluded that humans must have some way of recognizing these three primary colors. They created what is called the trichromatic theory of vision. They were the first to identify that our eyes have special receptors meant for these three primary colors. A man by the name of Thomas Young also made a similar discovery at about the same time. If you remember, we call this a multiple or a multiple event. Today, we know the theory as the Young-Helmholtz trichromatic theory of vision. But if you remember, a man by the name of James Maxwell actually helped Helmholtz create the initial theory. The co-founder, the co-author, the co-developer gets no credit. It is the young Helmholtz, not the Maxwell-Helmholtz theory. In the late 1800s, two German physicians, Gustav Fritz and Edward Hitzig, used electricity to map brain functions. In the late 1800s, the use of electricity in these animal studies became more refined. Two German physicians, Gustav Fritz and Edward Hitzig, used electricity to map brain functions, and their work was the first experimental evidence that the motor cortex is responsible for voluntary movement. That area of the brain is about right here. They restrained live dogs, cut away their skulls to expose their brains, and stimulated different parts of the cerebrum using batteries. When they touched one part of the motor cortex, the feet moved. When they touched another part, the tail moved. Another part, the tongue moved. They conducted these experiments on a variety of dogs of different ages and different breeds. They concluded that different parts of the motor cortex are associated with specific parts of the body. On the screen, you see an example of this in humans. Fritz and Hitzig studied animal brains. I wanted to show you an example of what they were trying to do. The information you see here comes decades later when we begin to experiment with human brains. Now that we've learned about decapitation studies and the animal studies, let's take a look at the nerve cell studies that were conducted in the 1800s and 1900s. The first microscope was invented in 1590 by a father-son duo, and researchers could see bacteria as early as 1675. Over the next 200 years, it would go through several revisions. You can see the 1847 version here on the screen. This is actually Charles Darwin's microscope that he used in the 1840s. These technological advances allowed researchers to see things that they had never seen before, including nerve cells. Camillo Golgi was an Italian pathologist who developed a special staining technique using silver nitrate. This technique allowed researchers to look at nerve cells under the microscope. In the image on the screen, you can see Golgi's actual illustration of the brain cells that he was able to see under the microscope. At about the same time, a Spanish pathologist and neuroscientist, Santiago Ramon y Cajal, suggested that there must be a space between these nerve cells. He also looked at them under the microscope. In 1906, they actually shared the Nobel Prize for establishing the neuron as the basic unit of the nervous system. Here you can see that Cajal's illustration of nerve cells is a little bit more detailed than Golgi's. Sir Charles Scott Sherrington was an English neurophysiologist. He was focused specifically on the functions of the mind, and he approached the study of this through physiology. He studied spinal dogs, dogs that had had some type of damage done to their spinal cords. Their reflexes tended to be more pronounced than those of intact dogs. So he concluded that the cortex had an inhibitory effect on reflexes. When the cortex was damaged in some way, those inhibitions were raised and dogs were more likely to spaz out whenever they should simply just turn their head or lift their ears. Sherrington published a book in 1906 to explain how the nervous system and the body coordinate to make the behaviors that we see every day. Reflexes, he said, are the simplest unit of this integration. Complex behaviors then are the result of these reflexes, these simple behaviors chained together, something he called reflex chaining. The synaptine, as he called it, we call it the synapse today. The synaptine is the site where these basic reflexes interact, join together, chain together to create complex behavior. Sherrington also proposed the idea of neurotransmitters, some chemical that allows neurons to communicate with one another. In 1932, he won a Nobel Prize with Edgar Adrian for their work on the functions of neurons. In this lecture, we reviewed some of the earliest studies looking at decapitated heads, animal brains, and nerve cells. All of these studies contributed to the emergence of psychology in the 1800s. In next week's lecture, we learn about German psychology and the father of modern psychology,